Hello everyone and welcome back to my channel. So today I am back with another episode of Read Robbins with me. Today I'm going to start the chapter of genetics from Robbins. Now before I start the chapter I want to give a few disclaimer. Now the chapter of genetics is having a lot of overlap with biochemistry, okay? And what I will suggest you is that see Robbins में जो disorders are explained here are given here, all the points are so important. You can't miss out on any of those points, and if you want to write a short note, I think these points are enough to understand. And also for your Neet PG exams, these points are super important. So if you do it from here, you can actually skip doing this from biochemistry as well. That is completely your take. But once you go through Robbins, I'm going to take you through Robbins. Once you do that, you will understand that so many important points are getting clarified here. Okay, that is one point. Number two is you will find the pedigree analysis from this chapter. So I have tried my best to explain the pedigree charts, because in the exam you will be given a chart and you have to diagnose what kind of disease or disorder that falls under. So you need to have your basics strong again, and this part is super important and also very much conceptual. Okay, and you can just do it from Robbins. You need not do anything from anywhere else for this part. Third point is that there are few topics here which I haven't addressed here. I'm going to do it in a separate video. The first one is familial hypercholesterolemia, and then the various genetic methods like the PCR, then FISH, then karyotyping, and your sequencing methods, especially the NGS. So these are the 4 to 5 topics which I haven't addressed. I'm going to post a separate video because I feel that if you do these topics exactly and entirely from Robbins, somewhere or the other, I feel that you need to have some extra inputs on these topics. Then it will be easy for you to understand. So some extra points should be added while reading these particular topics. So I will incorporate those points. I will make some review notes, and that will actually encompass and complete those topics. So doing those topics essentially from Robbins will not be that helpful. That's why I've skipped those topics, and I'm going to post a separate video in which I will inculcate this entire of all the other remaining topics. So with this note, now let's dive straight into Robbins. Now basically in this chapter of genetics, you need to understand there are three basic categories of genetic disorders. Number one is the single gene defect. Okay, disorders related to mutations in single genes with large effects. Number two is going to be your chromosomal disorders. Number three is going to be your complex multigenic disorders. Okay, so these are the basic outline of the genetic disorders. Now coming to the single genes, you must know that these are typically not present in the normal population, and they are highly penetrant, meaning the presence of this mutation is associated with the disease in a large population of individuals. So what is the meaning of the word penetrant? Penetrant means that if this mutation is present, then it will be present in a large population of the individuals, means the disease will be prevalent in a large number of people. So these are basically the Mendelian disorders, and along with the Mendelian disorders, mind you, there will also be non-Mendelian disorders coming under this single gene defects. Okay, so this is number one. These points, whatever I have highlighted, are all MCQs and are all important points. And I must tell you that this chapter is so important. All the points that I've highlighted, you can see that I've highlighted so much. I've underlined so much because I just can't miss out on any of these points. Okay. Anyway, so chromosomal disorders mean these are structural or numerical alteration in the autosomes and sex chromosomes. All these points are important. Each of these points makes sense, and they have got a very high penetrance. Okay, again an important point. Next is your complex multigenic disorders. Now what is this? This is basically a multifactorial. Okay, this is basically a multifactorial disorder. Why? Because you find that these are more common. There are interactions between multiple variant forms of genes and environmental factors. So not only the genetic factors, here the environmental factors also comes into play, and such variations in the genes are known as polymorphisms. So what is polymorphisms? Polymorphisms are variations in the genes within the population which gives rise to complex multigenic disorders, and they are all multifactorial. It is only when several such polymorphisms are present in an individual, hence the term multigenic or polygenic. So there have to be several such polymorphisms to be able to call it multigenic or polygenic. Okay, because each of this polymorphism has got a low penetrance. So only when there will be a large number of polymorphisms, only when there will be several such polymorphisms, then only it is going to have a high penetrance, and that will give rise to multigenic disorders or multifactorial disorders, as for example like atherosclerosis, diabetes, hypertension, autoimmune diseases, because you see these are all having so many other factors associated. It is not that a genetic factor is associated, a lot of other surrounding environmental, dietary, etc., so many other factors are also responsible for these diseases, and therefore they are called as complex multigenic disorders. Okay. Now, mutations, very, very important. Short notes are randomly asked from this chapter, from anywhere you can be asked. And of course, MCQs will, this is the basis of so many MCQs. So I just can't stress any more on the importance of these chapters and the importance of the points. I'm going to go through it. So mutation is defined as a permanent change in the DNA. Mutations that affect the germ cells are transmitted to progeny and give rise to inherited diseases. Fine. Now, what are the principles? You see, there are so many principles of mutation. Number one is the point mutations within the coding sequences. I will first mention the general principles and then I will explain you one by one. Number one is the point mutations within the coding sequences. Number two is the mutations within the non-coding sequences. I've already explained what is the meaning of non-coding sequences in the first chapter, right? So now you understand this. There is so much of correlation. We always tend to ignore the first chapter. इतना important भी नहीं है, it may be not that relevant chapter, but you see that basis of so many things are there, and this is how your concept building takes place. So once you know what is non-coding sequences, then only you'll be able to understand this much better, right? Next is the deletions and the insertions, and next is the alterations in the protein coding genes other than the mutations. Okay, mutations के अलावा भी there are different other alterations taking place. What are those alterations? Then alterations in the non-coding RNAs. This is also the same thing. Okay, now coming to this one by one, if you talk about the point mutations within the coding sequences, you see that a single base is substituted with a different base. Okay, single base is substituted with a different base because these mutations altered the meaning of the sequence of the encoded protein. They are termed as missense mutations. Now, this is having a classification. What is this? Number one is the missense mutation. Number two is the nonsense mutations. In the missense mutations, again, you have the conservative and the non-conservative. This is just the outline of the point mutation. It is just that one base pair is getting replaced by another base pair. So what's happening is, if you see that because of this point mutations, there is an alteration in the meaning of the codon, in the meaning of the sequence, that is called as missense mutations. Okay, and what is nonsense mutation? Nonsense means once the codon is replaced by another codon, there will be production of a stop codon, and whenever this stop codon will be formed, then the chain will be terminated. There will be no more further processing or translation. Okay, like for example, you can take the example of this, that a point mutation affecting the codon for glutamine or CAG creates a stop codon UAG. So here, if the U is substituted by C, then you get the codon which is a stop codon, and this leads to premature termination of beta globin gene translation, and this happens in beta 0 thalassemia, right? So since there is no globin production here, so there is beta 0 thalassemia. So thalassemia, this is a molecular basis of the formation of beta 0 thalassemia. When I will talk about thalassemia in the chapter of hematology, then you will see that this will also be an important molecular pathogenesis, right? This is how you should correlate. So now I was talking about, so this is all about the nonsense mutation, and now I was talking about the missense mutations. What you see here is that the new codon which is formed, or the new amino acid which is formed, if that is biochemically similar to the original one, then it is called as a conservative mutation. Means whatever mutation has happened, but still it is giving rise to an amino acid which is biochemically similar to the original one, which means because of the mutation there is not much difference. So that is why it is called as a conservative mutation. But when the mutation replaces a normal amino acid with a biochemically completely different amino acid, that completely changes the entire sequence, and that is how a different kind of an amino acid is generated, maybe the basis of a disorder, then it is called as a non-conservative missense mutation. So an example of this non-conservative mutation is the sickle cell mutation affecting the beta globin chain of hemoglobin. What happens here is that the nucleotide triplet CTG or GAG in mRNA, this CTG is coding for glutamic acid. and now it is getting converted to CAC. So basically the T here is getting converted to A, right? And now this CAC is coding for valine. So completely changed. The glutamic acid is being replaced by valine, and this, you know that this substitution is the basis for sickle cell anemia, right? So sickle cell anemia means this point mutation in which the glutamic acid coded by CTC is getting converted to valine coded by CAC, where there is replacement of the T by A. So okay, so I hope that you understood this, and this is the image showing the same thing, that there's a nonsense mutation where you can see that the C is getting converted to U, and there's a generation of a stop codon, and this is what is shown over here. So this was the first part. Now coming to the mutations within the non-coding sequences, you know that these non-coding sequences are what? These are basically the regulatory sequences which interfere with the binding of the transcription factor, and that's how they lead to a reduction or a total lack of transcription happening in thalassemia. So thalassemia, mind you, thalassemia has got so many molecular bases. This was number one that we studied here. This is number two basis. Why I'm saying this? Because molecular pathogenesis of thalassemia is again an important question. So once we come there, we will be able to correlate. We should be able to remember that we have read it here, okay? So number one, what's happening is that there are point mutations or deletions which are involving these regulatory sequences. And once this happens, this will interfere with the binding of the transcription factor. Once these factors are not able to bind, there's going to be marked reduction or a total lack of the transcription happening in thalassemia. Number two is that sometimes point mutations also happen within the introns, and that will lead to defective splicing of the intervening sequences. This will interfere with the normal processing of the mRNA, right? And that will cause a failure of the mature mRNA. The mRNA will not be able to process and work and function properly. Translation will not be able to take place, and the protein will not be synthesized, okay? So this is number one. So this is number one, and this is number two mechanism happening in these kind of mutations which is taking place within the non-coding sequences. These are again important points. The one I've highlighted in yellow is a point, and the one I've highlighted in blue is another point. So these are the 2 points by which you can understand that mutations can also happen in the non-coding sequences and how that actually affects. Because why this is important? See, because mutations happening in the codons which are giving rise to proteins or amino acids, mutations within them is giving rise to a disorder, and that is understandable. But mutations within the non-coding sequences can also give rise to a disorder. You understand? Non-coding sequences are not forming any translation, they are not forming any proteins, but in spite of that, the mutations happening within these sequences are actually causing so many kinds of other disorders. And that is why I said this concept of non-coding sequences is so much important, that I've already explained in the first chapter. Now coming to the deletions and insertions, yes, this is again an important point. You must understand that if the number of base pair is 3 or multiple of 3, then the reading frame will remain intact, okay? So if there is an insertion or deletion of 3 codons together, then the frame, the other part of the codon will remain the same, okay? And an abnormal protein lacking or gaining one or more amino acid will be synthesized. However, if the number of affected coding bases is not a multiple of 3, this will completely alter the reading frame, and that is what will be called as a frame shift mutation. Now I'm going to show you this by an example. If you see here, see, this is the cystic fibrosis wala DNA part. Now see here, 3 bases are getting deleted. Now once these 3 bases are getting deleted, the amino acid which is being generated, that is the phenylalanine, is no more generated here, and this one is deficient here, and because of which there is formation of cystic fibrosis DNA. But see the other parts are normal, which means this deletion is not getting transferred like this. The entire codon is not getting affected. This is not a frame shift mutation. But if you see this example, where you see that there is insertion of 4 codons, and once these 4 codons get inserted, you see the entire frame is shifted. You see each of these amino acids are different now, Hannah, because these codons are a multiple of 3. So whenever you insert or delete any codon which is a multiple of 3, then the entire frame will remain intact. But if there is insertion of 4 or 5 which is not 3 or not a multiple of 3, then these codon sequences will get automatically altered, and that will give rise to something called as frame shift mutation. The entire frame of the entire reading frame of the entire sequence is going to be altered. So this is what is happening in hexosaminidase A gene, leading to Tay-Sachs disease in the Ashkenazi Jews. Again, these epidemiological points are also important. So this is again an important example. So I hope you are able to understand this. These are very simple things, but you must have a proper grasp over it. These are very easy things. Once you know, you will never forget. Now, if you talk about the alterations apart from the mutations happening in the protein coding genes, they are the amplifications or the deletions or the translocation, okay, resulting in gain or loss of protein function. This amplification, deletion, translocations, for example, in the BCR ABL, in the CML. This is just an example. And if you talk about the non-coding RNAs, I've already explained this before, that these serve important regulatory functions, and these miRNAs and the long non-coding RNAs plays an important role here. This is a very important short note which is explained in the first chapter. Now comes another important part, and that is the trinucleotide-repeat mutations. This is amplification of a sequence of 3 nucleotides. Okay? So what's happening is 3 nucleotides, any 3 nucleotides is getting amplified. Okay, means repeated increase in the number of these particular 3 nucleotides. So what's happening is, whatever sequences are there, almost all the affected sequences, they share these nucleotides guanine and cytosine. These two nucleotides will always be present in whatever amplification takes place. The prototype is obviously the Fragile X syndrome, a very famous question. All the important points, I'm going to discuss it later in this chapter, are very, very important. So what's happening? Usually there is 29 repeats, but in this Fragile X syndrome, the repeats of CGG, okay, CGG, very, very important questions. It is repeated 250 to 4,000 times, okay, and in the gene known as familial mental retardation 1 or FMR1 gene, okay. And because of this, the normal expression of FMR gene is no more possible, because there are so many repeats of this gene, and this leads to intellectual disability. Another very important point is also a very important point. Another important point is that distinguishing feature of this trinucleotide repeat mutations is that they are dynamic. Very, very important question. That is, the degree of amplification increases during gametogenesis. With each generation, with each gametogenesis, the degree of amplification will keep rising. Means from 29 is going to be 300, then is going to be 500, there's going to be, let's say, 2000, then 4000. So with every successive generation and every successive gametogenesis process, this amplification will tend to increase, okay, and that is why they are called as dynamic. Very, very important point. I will talk about this when I will come to the details of this Fragile X syndrome. That is why we have something known as the pre-mutation, okay, which ultimately gets converted into a full blown mutation in the further process of gametogenesis. We'll talk about this later. Okay, so now coming to another point, and that is, you should not get confused with these words: hereditary, familial, and congenital. Hereditary and familial, these two words are used with having the same meaning in mind. So hereditary disorders are derived from one's parents and are transmitted in the germline through the generations, and therefore are familial. So these two words are interconnected. But congenital just means born with. So congenital has got no relation with something called as hereditary. Congenital means that they are just born with it. It may or may not be genetic. Okay, so don't get confused with these words. Congenital doesn't necessarily means that they will always have to be genetic. Okay, now coming to this Mendelian disorders, very, very important, because here we are going to learn the different kinds of patterns and the pedigrees. Okay, now, obviously, this is the result of mutations in the single genes having large effects. Mutations in the autosomal genes produce partial expression in the heterozygote. So, heterozygote may there is partial expression, and homozygote may there is full expression. Like, for example, sickle cell anemia, if you see, then you know that it is because of substitution of the hemoglobin A by hemoglobin S. But listen to this very carefully. When the individual is homozygous, what I've said here, that homozygous means there is full expression. So when the individual is homozygous for the hemoglobin S, then all the hemoglobin is abnormal of the HbS type, and even with the normal saturation of oxygen, the disorder is fully expressed. But if it is a heterozygote, then only a small proportion of the hemoglobin is HbS, means they have HbS and they also have HbA, because they are heterozygote, and therefore this red sickling occurs only under unusual circumstances and it doesn't occur at normal circumstances. That means unusual circumstances means when there is exposure to lower oxygen tension, and this is called as a sickle cell trait. So sickle cell trait is happening when it is a heterozygote, and full blown sickle cell anemia is happening when it is a homozygote. Okay? Now, so Mendelian traits can be dominant and recessive, but in some cases, both of the alleles of the gene, they are contributing to a phenotype which is called as the codominance. Means both the alleles are expressed in the similar way. Like you have the ABO system, right, where you can see that A and B blood group, the A is also expressed, the B is also expressed. So that is codominance. Now these points are important to understand, the meaning is important to understand. So what is pleiotropism? Pleiotropism means a single mutant gene may lead to many effects. Okay? For example, if you talk about sickle cell anemia, the mutation is one, but you see that it is giving rise to hemolysis. At the same time, there's also occurrence of splenic fibrosis, organ infarcts, bone changes. So a single mutant gene is present, but it's having so many end effects. This is called as pleiotropism. Example is sickle cell anemia. What is genetic heterogeneity? Mutations at several genetic loci may produce the same trait. So mutations are happening at several positions, but they are having the same effect. So in genetic heterogeneity, the effect is the same, whereas in pleiotropism, the effects are different and the mutation is one. Okay.